Excuse me, may I ask a question? I think it's the third, or in the slide about carbohydrates, there are information that it is an energy source and energy store, and there are a graphic that when glucose plus oxygen turn into CO2 and water, and energy ATP. What is energy ATP is exactly?
Okay, so ATP is the high energetic molecule, so it contains energy in its bonds that could be extracted very easily. And usually enzymes, which are catalyzers, they use these molecules to fuel their work, which means to perform these chemical reactions, they use the energy that is stored within these ATP bonds. And these ATP bonds are formed during the process of respiration.
Okay, thank you.
Mm-hmm. So do you see my screen?
Yes.
Okay, so sugar, for example, the glucose, it has 6 bonds between these carbons. And whenever we have this process of respiration, which means we oxidize the glucose, we break these bonds, okay? And this energy actually needs to get somewhere. So some of this energy is being irradiated as heat. So we will have 6 CO2 plus 6 waters, plus some heat and plus energy that mitochondria could catch and put it in the ATP. So energy stored in ATP. This is high energetic, very easily giving this energy molecule, and that's why it's kind of unstable. So whenever we have ATP, we need to use it almost at the same time. So, for example, we have some kind of enzyme that takes substrate 1 and substrate 2 to produce the product. But to make the distance between these two as small as possible, it needs to kind of change its conformation. And whenever its conformation changed, it means that it produced some work. And to work, every enzyme needs energy, and it takes it from ATP. So it takes it from ATP, becomes ADP. So this ATP converts to ADP, but the energy stored in one of these bonds is given to this enzyme. So it performs its job.
And so ATP is just one type of molecule, or it can be in different carbohydrates can have ATP?
Okay, so ATP is the universal storage of easily accessible energy, and it's adenosine triphosphate. And if you take adenosine, it's just the, as you know, there is a 5 carbon carbohydrate bound to the phosphate group. And on the other side, it's bound to the nucleotide, in this case, adenine. So this is ribose. And here is phosphate. So this monomer is called AMP, so adenosine monophosphate. Whenever we have another phosphate here, it becomes ADP. And when we add the third phosphate, it becomes ATP. And when it became ATP, it means that a lot of energy stored in these two bonds. So energy. And whenever we break these bonds, we can use this energy, either this or two, and use it in the enzyme catalysis. So this is very general, very kind of usual molecule that is being used. We can also have GTP, CTP, and I guess it's usually 3 molecules that are used for energy, but the most abundant one is adenosine triphosphate, or ADP. Is it clear?
Yes, it is. Thank you.
Mm-hmm. We just take any sugar, and some of its energy goes to the production of ATP.
I have one question regarding DNA and RNA. It is not clear for me where are DNA and RNA located? Are they located in every cell of the body, or, for example, in human body, are they located in just one specific organ? Or could you please clarify this?
So every single cell that you can imagine must include either RNA or DNA. In most cases, for all cells, you will have DNA inside it to store the genetical information, and RNA as the kind of housekeeping nucleic acid. So there are few cell types within human's body that do not contain DNA, and one of them is red blood cells, erythrocytes, and they do not contain this DNA because after they mature, they need to kind of reduce the amount of non-needed organelles in their body so that they can perform their very specific job most efficiently. So if you think about the erythrocyte, so it starts with one of the progenitors of blood cells. And as any cell, it contains some kind of nucleus. But after the process of maturation, it becomes this discoid. So if you look at it from the side, or you can also see it like this. And it does not contain any organelles that kind of reduce the internal space of the erythrocyte. So this whole space is occupied by the hemoglobin. So this is very specified, or it is also called differentiated cell that is being used for a very specific role in the organism. So it only contains hemoglobin. So if you think about the same size of the cell, it will look like this because of this nucleus. So this is erythrocyte, and this is usual cell. So as you can see, we don't need this huge weight that we need to carry, so we just get rid of it. That's why during this maturation, the red blood cell loses this nucleus. And this is one of very few cell types that doesn't have the DNA. But in general, just think, for example, about bacteria. So they do have DNA because they are isolated organisms, unicellular organism. It really needs to perform some actions, perform some chemical reactions to live. So to live, it needs proteins. Not only proteins, everything actually, but most of the genes, they code for proteins. So these proteins, they make it possible to breathe, eat, and divide, for example, for these small bacteria. That's why it really needs to contain these circular DNA. Also, whenever it divides, it needs to kind of give these DNA, these traits that are encoded in DNA to its daughter cells. So whenever it divides, we have two bacterians with the same genetic information. So it really needs to copy this information and then give it to its daughters. And if you take, let's say, human body, any cell, it also needs to divide. It needs to perform its specific functions. So again, functions and division. For everything that is within these limits, we need to produce, constantly produce proteins, etc., etc. That's why they need to be encoded in the DNA and then produced. So first you have DNA. You have a copy or blueprint from this DNA of one gene, and it's stored in mRNA. And then this mRNA is sent to ribosome. And this ribosome translates this information encoded in mRNA into protein. That's why we really need this DNA and RNA to just function as a cell or as an organism. Alejan. So,
yes.
Yeah.
Okay, so here on this part of the image, you can see the nucleotide. So do you see my mouse?
Yes.
Yeah.
Okay, cool. So this is the phosphate. This is the 5-carbon sugar, and this is the nitrogenous base. So as you can see, it's covalently bound to the sugar as well as the phosphate. And then to make this chain like this, it really needs to bind covalently. This phosphate needs to bind to another sugar. And if you start counting carbons from this O, it will be 1, 2, 3, 4, 5. So the direction, for example, of the nucleic acid goes usually 5', 3', and that means it goes up, down. And in this case, the first nucleotide will be the upper one.
I just want to know what's the connection between deoxyribose and DNA? Like what's
The thing is, this ribose, deoxyribose, it is the part of DNA molecule. It's not connected, it's just a part of it. And because this part exists in the DNA molecule, we have this huge chain. We have this sugar called deoxyribose. And here you have OH, 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 and OH. So this is just a sugar, cyclic sugar. If you think about it, inside the molecule of DNA, it needs to bind in the position 1. So this is 1, 2, 3, 4, and 5. In the position 1, it needs to bind to the nitrogenous base. So we remove this hydrogen and have, for example, adenine here, whichever adenine, thymine, cytosine, or guanine. And also we need to add one phosphate here. So remove these hydrogen and add phosphate. So it will look like O, OH, and OH. And this has become a nucleotide. And to make a chain of it, we remove one of these hydrogens and connect it to the next third carbon of the next deoxyribose. Okay, so here we have another nucleotide that is bound to O and then to directly bound to this O. So phosphate here and adenine here. So adenine here. And this is how it binds to another nucleotide and makes this chain. And within this molecule, we have deoxyribose, and it plays a crucial role.
There is one more question. In the structure of sugar, there is a thicker bonds like bold and thinner. What's the difference between them?
It's usually just the representation. So whenever you have corners, it means that we have carbon there. So this corner means carbon, 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 and carbon. These thicker lines, they just kind of represent, it doesn't make kind of difference if you show it with thicker lines or with usual ones. In this case, I guess it just looks more 3D or 3D projectional. It will be the same as showing it like this.
And may I ask, well, as I know, there are a hydrogen bond between the bases of DNA, like AT, CG, or
Yes.
it's not. It is, yes?
There is a hydrogen bond. Well, actually, there are two hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between cytosine and guanine.
Yeah, hello, about proteins. It was said that amino acid sequence should be exact and is dictated by nucleotide sequence of the genes. What is nucleotide sequence, or how to understand that sentence?
Okay, so this sequence, for example, A, T, C, G, G, C, C, A, T. Whenever you see something like this, it means in the DNA, in one part of the DNA, you have adenine, thymine, cytosine, etc., etc. And let's say that this is the part of the gene, okay? They're usually much bigger in the length. So every 3 nucleotides, so this is called nucleotide within the DNA. And every 3 nucleotides, they form so-called codon. And each codon encodes some kind of amino acid. So when we have 3 nucleotides, if we start reading them with a special table that shows which codons mean which amino acids, you can actually say the sequence of amino acid according to the sequence of nucleotides. So I'm not really sure the real meanings of these codons, but let's say this is alanine, this is lysine, and this is leucine. And whenever we have the initial codon sequence or initial nucleotide sequence, we can predict which proteins we can get from this sequence, because we know that these particular codons, they encode these particular amino acids. So again, whenever we have DNA, so this is DNA, and it encodes protein gene. So this is the blueprint of the protein, okay? So through the process of RNA polymerization, or so-called transcription of the RNA, we make a copy of this gene with the RNA nucleotide. So we make this copy, and after being processed, it will be called messenger RNA. And this messenger RNA is the algorithm for ribosomes. So you have 2 units of ribosome, and this mRNA will go here between these 2 ribosomes. And they will read it codon by codon. So let's say these are codons on this mRNA, and they are the same as the codons on DNA. And every single codon means amino acid. So this ribosome, it recognizes these codons and starts, so it takes amino acids that are floating inside the cytosol or cytoplasm, and it takes them and starts building the protein sequence or protein chain, so-called polypeptide. So it goes from one side to another, reads every single codon, and adds this related amino acid to the peptide chain. And when it has finished the work, this polypeptide kind of floats away and being produced further. So the protein starts its maturation. And that's how the sequence of the protein is related to the codons and the DNA.
Yeah, so like, for example, 3 nucleotides, it's like 1 codon, right? And
Yes.
it forms like amino acid?
It doesn't form, it codes for 1. So let's say you're a special agent, and from FBI, you have 1 dictionary, okay? And in this dictionary, it says that 15A3 means hello. For example, 23A3 means do stuff. And you can recognize these digits as something that you can interpret with this dictionary, okay? And because you have this dictionary, you can have a sequence of these numbers, etc., etc. And then, because you know what's the meaning of each of these ones, you can build a whole sentence and receive the instructions, okay? It's almost the same. So the ribosome knows these codons, so CAT, GGC, ATC, and it reads them and it builds the protein according to these encoded pieces. In our case, every piece means a word. In their case, every codon means one amino acid.
Yeah, and I want to clarify. So transcription, it's like reading these codons, right? And translation is like forming polypeptides, right?
Almost there. So.
And are there any other like functions of RNA and DNA?
So let's say you have DNA, and here is a gene. You cannot use this DNA because it's really huge. Yeah, it's a bank of all genes, okay? So to use it, you need to make a copy. And you make a copy of it with the mRNA production. So this is DNA. And this process of copying is called transcription. And if you look here, script means write. So it's coming from Latin. It means write, and transcription means write somewhere. In this case, you take DNA and you write into RNA. Okay. So the copying from DNA to RNA is called transcription. And when you have this information in the language of nucleic acids, you need to somehow translate it into language of proteins. And for that, you need a translator, the so-called dictionary, or you can find a lot of terms to name it. Okay, and here they are not the same. So this translator is the ribosome, because it knows the meaning of one word, one codon, and it can translate it into the language of amino acids. So this is translator. You have a piece of information in one language and you receive this same piece in the other language. And this means translation. Okay, so replication of DNA is needed when we have a cell and it performs the division. So this cell will give two daughter cells. So daughter cell 1 and daughter cell 2. And usually you really need to give these both daughter cells the same genetic information. Otherwise you will get something like cancer in the end. So these genetic information should be identical for both of the cells. But how you do that? You actually you have only one copy of all DNA that you have. So you just replicate it. So instead of one DNA molecule, you will have two the same ones. You made a copy of it, and this is called replication. So you make a replica of the initial DNA. It's also DNA, and when the time comes, special process kind of divides the genetical information and then divides the cell itself on the same region. So you will have two separate cells with the same amount of DNA, and this DNA is identical. And this happens in all organisms. So if you take your neurons, their DNA will be identical to your skin fibroblasts, for example. And this is the same DNA.